So in today's lesson, we're going to be learning about photosystems. So as you learn, a photosystem is a critical component of the light reactions. It's a structure that is essentially a light gathering molecule. And so we're going to talk to you about that prior to beginning the details of the light reaction. So you'll remember from our previous lesson that photosynthesis is divided into two distinct phases. The first of which is the light reactions, and the second is the light independent reaction, or the Calvin cycle. So we can divide the light reaction into three distinct components. The first is what we call photoexcitation. So photo meaning light, and excitation, well, what would be excited? We're talking about a photon being absorbed by an electron of chlorophyll. And so I'm going to discuss that even further in the next segment. The second component of the light reaction is an electron transport chain. So you'll remember that overall, the function of the light reaction is to do what? Well, it is to produce ATP and NADPH that's going to fuel our Calvin cycle. And so how the ATP is going to be produced is via an electron transport chain, in a similar manner to what we would have seen in cellular respiration. So we're going to see an electron transport chain that's going to grab onto that excited electron that's produced via photoexcitation, and it's going to travel through a sequence of membrane-bound electron carriers. It's going to just travel through that. That's going to create a hydrogen ion reservoir, again, similar to what we saw happen in cellular respiration. And then finally, in a similar manner to cellular respiration, there's going to be a chemiosmotic process by which a proton motive force is going to allow us to synthesize ATP using the enzyme ATP synthase. So that is basically the summary of what we're going to see in today's lesson. So I want to start us off by looking at a diagram that's representing photoexcitation as a process. So you'll remember from grade 9, grade 10, science, chemistry in particular, that we have protons and neutrons in our nucleus of our atom, and then we have electrons just sort of in distinct energy levels surrounding the nucleus. And from grade 10 optics, you'll remember that if we bombard an electron with enough energy, then what is going to happen to that electron? Well, typically what we will see is that electron will get promoted to a higher energy level. And so that is what's essentially happening during photoexcitation. We're seeing electrons in our primary pigment, chlorophyll A, absorbing energy from light and using that light to promote electrons into a higher orbit. Now, the thing that happens here in our chlorophyll molecule is, instead of that electron being, eventually coming down to ground level, what we're going to see is that electron is going to be accepted by something that we call a PEA, or a primary electron acceptor. And from there, what's going to happen to that electron, the fate of that electron, is that it's eventually going to move into our electron transport chain, but we're going to talk about that later. Okay, so that's what I want you to understand right now, the idea of photoexcitation, which is that, the sun's energy is going to come in, and that energy is going to be used to excite an electron in our chlorophyll molecule, and that electron is going to move to a higher orbit and get grabbed by a primary electron acceptor. And that is essentially what's happening. Now, as a result of that transfer, we might see some heat loss because no transfer is 100% efficient. And we might see a little bit of fluorescence because not all of the energy may be accepted by that primary electron acceptor. And so if we were to actually look at chlorophyll under a UV light, we would see that it would appear sort of red. And that's symbolized by this red little line here in that photon. So that fluorescence it would be giving off would be sort of red in color. So I just took a minute to search out on my browser a picture of this. So here is, some chlorophyll extract, and here's that same chlorophyll extract that we see under UV light, and that is because, our UV light bombards it. That will excite some electrons, and it will give off that fluorescent sort of that red color as a result. So now let's take a minute to write some of this down. So what was happening? Well, a photon of light is going to strike our chlorophyll molecule. And where is that located? Well, we've talked about this. This really is just in our thylakoid membrane. We're zooming in on that phospholipid bilayer, and we know that our chlorophyll molecules are attached to the thylakoid via that phytyl tail in the chlorophyll molecule. We know that before that, those electrons are going to be in what we refer to as the ground state. That just means they're in their sort of everyday position where they would be with their lowest potential energy. But we know that as soon as that photon hits our chlorophyll molecule, that's going to cause that electron to be promoted to a higher energy level. That is what we refer to as photoexcitation. That there is the photoexcitation. Okay, but that what's happening to that electron? Well, it's going to be grabbed by our primary electron acceptor molecule. And that's going to allow it to shuttle off to an electron transport chain. And, I didn't talk about this before, but we can consider that a redox reaction of sorts, because we know that chlorophyll is losing an electron and our PEA is gaining electron. So we could say that our chlorophyll is oxidized, and I'm just going to remind you real quick about Leo-Ger. So Leo, loss of electrons is oxidation, and gaining of electrons is reduction. So our chlorophyll molecule has lost electrons, so it's been oxidized. Our PEA has gained an electron, so it is reduced. There we have it. So that is photoexcitation in a nutshell. Okay, so now down to the structure of a photosystem. So I want to orient you a bit. So in the last segment, we talked about chlorophyll A and photoexcitation. So really what we saw in the last little diagram where we saw, an electron from chlorophyll A being promoted to the PEA, really all they're doing is they're showing the PEA being sort of just adjacent to it, but you can still see that electron transfer. So that's just in our frame of reference for our photosystem relative to the last diagram. You'll also remember that in the last couple of lessons, we talked about how chlorophyll A is not the only light-absorbing pigment. And we have a broad range of pigments that will allow for light absorption at a variety of wavelengths within the visible spectrum. And that just allows us to make the most efficient use of that visible spectrum and its wavelengths of light. Because, of course, chlorophyll A has an absorption spectrum that is fairly narrow, right? So by having a variety of other photosynthetic pigments, we can capture more of light's wavelengths and make the most use of it. So in a photosystem, you're going to have what's called a reaction center. And that reaction center is always chlorophyll A. Chlorophyll A is, again, that main photosynthetic pigment. But in a photosystem, what you're also going to see is other pigments which are called antenna pigments. So you're going to have a variety of other pigments that make up the photosystem that are, again, just embedded in our thylakoid membrane. So this would be representative of our thylakoid membrane and the molecules embedded within it. So what is really happening is we have all these other pigments that are there that are referred to as antenna pigments, along with our other accessory pigments, and they essentially just channel energy towards our main reaction center, chlorophyll A. So they grab onto the energy from light in those wavelengths that chlorophyll A cannot absorb directly, and they take it and they transfer it to our chlorophyll A molecule. And that allows chlorophyll A to still make use of the other wavelengths. They eventually get to chlorophyll A, and then that energy is used to, again, promote electrons and then transfer them to our primary electron acceptor. So you can sort of think of that antenna complex here surrounding chlorophyll A as being similar to, and you guys are probably too young to remember this, but so maybe somebody in your world has talked to you about how, you know, old TVs or even radios for that matter might have like an antenna on them. And what's the job of that antenna? Well, the antenna is to take in sort of maybe radio waves and transfer them to the radio so that it can capture the signal, and then you'll hear some sound, right? So same idea with our photosystem. The antenna complex's job is to basically take in certain wavelengths of light and transfer it to chlorophyll A so that chlorophyll A can make use of it. So let's make some notes about this so that we have it to look back on. So first of all, our photosystems are going to act as that primary light harvesting unit of the chloroplast. So it's not just chlorophyll A working alone. We have all these other pigments helping with that. So as I said, it's composed of our reaction center, which is chlorophyll A, as well as that antenna complex, which is made up of all those accessory pigments that we spoke about. So I want you to take a minute to think about what the antenna complex is in terms of its structural components and what its function is before we move on. So I'm hoping what you thought about was that the antenna complex is going to be composed of a bunch of different chlorophyll molecules and all of those accessory pigments that we discussed. So for example, the carotenoids, the xanthophylls, and so on and so forth. And they're going to be embedded in that thylakoid membrane. And their job is to basically absorb and transfer energy from pigment to pigment until it eventually reaches that reaction center. Take a minute to see if you're on the right track with thinking about the reaction center. So again, what is it composed of, and what is its function? So I'm hoping that when you thought about the reaction center, immediately chlorophyll A came to mind, that primary light harvesting molecule. And basically what its job is to become photoexcited, to grab onto the electron that is being transferred to it via those other antenna parts of the antenna complex, and that will allow for an electron to be transferred to that primary electron acceptor. So we're at the stage in the lesson where I need to point out to you that, you know, there are two types of photosystems that we see in our electron transport chain. Because remember that primary electron acceptor is going to transfer an electron, our chlorophyll molecule is going to transfer an electron to our primary electron acceptor. And then that's going to be part of a bigger electron transport chain that eventually is going to yield us some ATP for the Calvin cycle. So, you know, just to orient you, to give you an idea about what we're talking about here is, you know, here's our thylakoid membrane. And then embedded in there, we're going to have our photosystem. And so along the line, we're going to have another photosystem. And I'm just making this shorter just for the sake of, you know, paring it down to the basics. And they're both embedded in that thylakoid membrane. And they each have a similar job to do. There are some variations, but that's a part of our next lesson. What you need to know, and this is, you're going to love this, photosystem 2 actually happens first, and photosystem 1 happens second if we're looking at this from a sequence perspective. And the reason for that is mainly because Photosystem 1 was discovered first, and then Photosystem 2 was discovered, so hence the name. But you do need to know that in the sequence of events, Photosystem 2 is going to happen first. So, they're both embedded in that thylakoid membrane. They differ in terms of the wavelength of light that they function best at. And so we're going to talk about that actually in the next lesson to greater degree, but I'm just going to point it out to you today. Photosystem 1 will operate best at 700 nanometers. That's where you're going to see chlorophyll A working its best. So photosystem 2 is best at operating at and absorbing wavelengths of light at 680 nanometers. So photosystem 2, for that reason, is often referred to as instead just P680. And photosystem 1 is often referred to as P700. So those are just names given based on the wavelengths of light that chlorophyll A is absorbing light the best at. And those are details that we're going to talk about in greater detail when we look at our next lesson. But the main reason for that happening is we know that small little structural differences can potentially make a really big difference when we're talking about biochemistry and biology in general. So, the difference in that slight difference in the wavelength of absorption is due to just a slight difference in the protein complexes that are involved in each of those photosystems. So stay tuned for tomorrow when we look at specifically what is going on in these photosystems, how is that all working together. But those are the basic structures of photosystems and how they function. So as always, reach out for help if you need it and for any points of clarification. I'm hoping that made sense, but if there's anything that's unclear or that you want more detail around, be sure to reach out.